this is very important for continuations of life. Yeah. So fertilization is a very, very important process, okay, in the developmental biology, because only by fertilization you have the zygote and further develop into the embryos, and slowly they become the full organism. So it is extremely important processes, okay, and it's quite complicated, okay, for fertilization. Okay, and after the end of this unit, then I'll show you a short video about human fertilizations. But before that, let's look at what is the importance of gametogenesis and fertilizations. So if we look at the research into how eggs and sperm are created, can help us actually to understand infertility, especially in humans. Okay, which affect up to 25% of the couples in Europe. 25%, that's mean in five pair of couples, there will be one piece which is infertile. So this is very high number. Okay, and it's also showed that the humans male infertility rate actually is going up, especially in China, and they are actually looking into the cause. What is the causative factors that actually contribute to the infertility of male? Okay, and in women, the infertility can result from a premature menopause, which is the premature ovarian failure, whereby the ovarians or the eggs are failed to form, a failure to ovulate, there is no ovulations, or polycystic ovarian syndrome, or what we call the PCOS, or the recurrence miscarriage. That's why that's mean the miscarriage is going on from time to time. So there is no implantations. Or when it is the implantations and the baby will not stick, and there are no healthy baby will be born. Okay, so this is happening, what we call the recurrence. And in men, the problems as stages of spermatogenesis contribute to infertility. And although estimates suggest up to 6,000 genes that might be involved in spermatogenesis, as yet only a few individual genes mutation has been linked to male infertility. And in pre-menopausal humans, women, or in adult men's sex organs are the major site of the steroid hormones and manufactures. And hormones actually play a very, very major role in gametogenesis. Okay, be it the male hormones or female hormone. Yeah. So these steroids, which is like androgens and estrogens, are not only essential for the formations of egg and also sperm, but they also have an important impact on the functions of other parts of the reproductive system, such as the endometrium, which is the lining of the womb in the woman, and also the prostate in men. So in addition, the steroids made in the ovaries and testes will circulate around the body, and it will affect the functions of the heart, the brains, and also the skeletons and the immune system, and can also contribute to the formations of the cancer in tissues, including the breast and also the prostate. And what are the health targets is that involve the infertility, premature ovary failure, the polycystic hormones, the ovarian syndrome, recurrent miscarriage, and so cancer. So it is very important for us to understand the gametogenesis and fertilization, okay, in animals, especially in humans. So what are the research activities such as the investigations at the Human Reproductive Sciences unit into the formations of the female gametes are aiming to address three different questions. First question is, how do ovarian follicles form, and what factors that actually influence the number of the follicles a woman form during her lifetime? And how is the formations of the new blood vessel, which is the angiogenesis in ovarian follicle regulated, and how is the follicle growth and death regulated? Okay, these are the very major questions that scientists are paying attention to understand the human's fertility. And the process whereby two sex cells, gametes, which are fused together and create a new individual, is what we call the fertilization. Okay, with the genetics potential derived from the both parents. So there are four major steps that involve in fertilization. Okay, the first one is the contact between the egg and also the sperm. That is the first step. Therefore, one of the four major steps. The second one, after they contact, then there are entry of the sperm into the egg. That is the second step. After the entry of the sperm and the egg, then there will be the fusions of the nuclei of the egg and the gene of the eggs, and that is what we call the fusions of the nuclei. Okay, and restorations of the diploid genome, and there is also an activations of the egg metabolism after the fusions of the nuclei of from the eggs and also the sperm. Then the activations of the metabolism will start. Okay, and they are accomplished four things upon the fertilization. First, the restorations of the diploid genomes, because the egg is an end, the sperm also an end, the genome of the egg and also the sperm. So the end and end that become two ends, become diploid. So there is a restorations of the diploid genome. Second is a species-specific sperm egg binding. This is very important because the species will not be like crossover. Okay, for example, the man cannot breed with cow or cannot breed with a monkey. Okay, because this is species specific. This is very clearly happen in the river or in the oceans or in the lake, whereby if you look at there are so many organisms in the river, in the lake or in the oceans, but how come the egg and also the sperm, they can bind together, they can make the baby? Because a lot of these organisms, they are external fertilizations. Yeah, so this is because of the species specific sperm binding processes. Okay, and the third one is egg activations, whereby after the fusions of the nuclei from the sperm and egg, then the egg will be activated and contributions of the paternal chromosomal centrosome material by the sperm. So there are four, right? So of course they are quite complicated processes, but we just need to know this. This is enough for you at this stage. Okay. So far, any problem or any questions before I proceed to the next slide?
No.
Is that clear?
No, doctor. Clear, doctor.
Clear, doctor.
Clear, doctor.
So see, this is the structure of the sperm. Okay, so each sperm consists of the haploid nucleus, a compulsion system to move the nucleus, and a sac of the enzyme that enable the nucleus to enter the egg, which is at the this is at the acrosomal vesicle. Okay, this contains the enzyme which will lyse the membranes of the egg later. Okay, if you look, this is the how the egg the sperm is formed. Okay, from there you have the nucleus, you have the Golgi apparatus, the centriole, and this is the mitochondria. Of course, mitochondria is very important. It's needed. Okay, and they are actually concentrated at the neck, whereby the mitochondria will provide the energy for the tail to for the propulsion, for the energy, for the tail to move. Yeah, so this is extremely important. So the centriole produces a long flagellum that will be the posterior end of the sperm, and the Golgi apparatus from the acrosomal vehicle at the future anterior ends. And the mitochondria, which is the hollow dots, will collect around the flagellum near the base of the haploid nucleus, and it will become incorporated into the midpiece of the sperm. This is the midpiece. All right, so they are concentrated here, and so the remaining cytoplasm is gestation and the nucleus condenses. Then the size of the major sperm has been enlarged relatively to the of course this is enlarged, as you can see, right? So if you look at the sperm, we classify them into the head, the sperm head, which is consists of the nucleus and also the acrosomal vehicle. And the neck, we have the neck here, and we have the midpiece, which they have a lot of azonim, and also the mitochondria, and which is the tail and also the end piece. Okay, so the tail is getting the energy generated from the mitochondria for the propulsion, so that they can move around. Okay, so it is very important. If the sperm cannot move, so there is no chance for them to meet with the egg, and there is no it is impossible for the fertilization to take place. Okay, So if you look at the structure of the sperm, we will do the transverse section here. Okay, and this is how it looks like under the electron microscope. And if you enlarge it here, you can see this is the adenine. Okay, this is the tail. Yeah, this is the tail piece. Yeah, this is the adenine. And if you look at the outer layer here, we have the plasma membranes, and we have this one what we call the radius spoke. Okay, and in the then we have also the nixin, which is the green, which is the green color here. And in the spoke head here, if you look at this is the spoke head, and in the spoke head here you also have what we call the subfiber A and subfiber B, and you also have this what we call the central singlets of microtubules here. Okay, and also you have the inner Dianne's arm and outer Dianne's arm. Okay, and if we enlarge the microtubule A here, then you can see you have the what we call the beta tubulins monomer and alpha tubulins monomer. Okay, this is very important for them to move around. Okay, so that they're helping the tail to move around, right? And this is the structure of the egg. And if you look at the egg, we have a very of course the egg structure is much larger than the sperm, and it is consists of the outer jelly layer. This jelly layer is very thick. It is very important to help to protect the nucleus. You see, nucleus is the innermost structure which is protected by the jelly layer and also the ventellin envelope and the plasma membranes of the nucleus. Okay, and cortical granule, and as you can see, the nucleus is embedded right inside here. And the halide structure here, it is the sperm. If you look at this one, yeah. So there are millions of the sperm which attached to the egg, and they are very actively to protrude right into, and everyone wanted to meet with the nucleus, but only one egg, only one sperm is successful. Only one sperm, yeah, will fuse with the nucleus, and after that, all the sperm will be disqualified by a mechanism, or I show you later. Okay, so this is the structure of the sea urchin egg during the fertilizations, and the drawing also show the relative size of the eggs and also the sperm. Okay, if you look at this one, yeah, this is taken under microscope, and you can see this is the head of the sperm, and this is the whole egg, which is like surrounded by the sperm. Okay. The B is a scanning of the electron micrograph of the sea urchin sperm found by the ventellin envelope of an egg. All right. And if you look at the structure of the hamster egg, they are slightly different. The hamster egg immediately before fertilization. So if you look at the A, this is the A, this is a hamster egg, or the ovum is encased in the zona pellucida, and this in turn is surrounded by the cell of the calamus. Polypodies are produced during the meiosis, is also visible during the zona pellucida. Okay, B is at the lower magnifications. Mouse oocyte is shown around by the collider carbon particles. Okay, we hardly see the Indian in here. Okay, because this is black and white, and it's included by the hyaluronidase matrix. Okay, but anyway, you can see this is the egg. Yeah, this is the egg at the lower magnification, but if we enlarge it, you can see something like this. Okay, so if you look at the eggs, all the materials necessary for the beginning of the growth and development must be stored in the mature egg, and that is why the size of the egg is much larger compared to the sperm, because they need to store a lot of information and materials for the very beginning of the formations of the zygote right after the fertilizations. But in contrast to sperm, the developing egg, which is the oocyte, not only conserve its material, but it's also actively accumulating more. And the eggs called cytoplasm, they have like a storehouse where they store, which includes the protein, the ribosome and tRNA, the messenger RNA, the morphogenesis factors and protective chemicals, and this is fine. Okay, well, we can always see that the egg is much larger compared to the sperm. right? So far, okay. Can you follow me?
Yes,
Okay,
doctor.
doctor.
Yes, doctor.
Right. So the recognitions of the egg and sperm is very species specific. Okay, so it is the interactions of the sperm and also egg generally proceeds according to the 5 basic steps. What are these 5 basic steps? Is first includes the chemo attractions of the sperm to the egg by soluble molecule secreted by the egg. You just imagine those egg of the fish or prawns, you know, they are in the oceans or in the lake or in the river, and you need to imagine that the oceans, they are have lots of buoyancy, the waters are moving around, and some could be very rapid, okay, like in the river and also in the oceans, okay. So how actually these sperm and egg can attract to each other? This is a question that's for you to ponder. Yeah, and also the exocytosis of the acrosomal vehicle to release the enzyme. Okay, the next one is the binding of the sperm to the extracellular envelope, which is the ventral layer or zona pellucida of the eggs. Then after that, it's the passing of the sperm through these extracellular envelopes. And lastly and finally is the fusions of the egg and the sperm cell plasma membrane. Only by fusions of the egg nuclei and or sperm nuclei, then we call the successful fertilization. Okay, so this is the basic. This is the basics 5 basic step. Okay, basics 5 basic step of the recognitions of the egg and also the sperm. Right, they have to go through all 5 these before the fertilization can successfully take place. Okay. Now, if you look at this one, this is the sea urchins and this is the mouse. They are slightly different. Okay, you can see that. Okay, what are the differences? First, if you look at the zona pellucida, in mouse, it is much thicker compared to the sea urchin. All right, can you see the difference?
Yes.
Okay, okay, and if you look at the process is very similar. Okay, first is the sperm contact the jelly layer. The jelly layer, of course, is the outer layer or the extracellular cord of the egg. Okay, so they are contact, right? So after that, if you look at sea urchin, the acrosomal reactions, yeah, the acrosomal reaction whereby they go through the jelly layer, at the same time they will digest the jelly layer, yeah, digest the jelly layer, then they bind to the ventellin envelopes. Then once they bind to the ventellin envelopes, then there are fusions of the acromo process membranes and egg membranes. Okay, but if you look at in the mouse, the sperm bind to the zona pellucida, and there's acromal reactions, and the sperm lies a hole in the zona pellucida. Right? It's different, right? But only they meet, the sperm meets the egg membrane, then they will release the nucleus of the sperm, so that the fusions of the nuclei from the egg and also the sperm can take place. That's slightly different in between the recognitions of the egg and sperm in the sea urchins and also the mouse, in the process of for them to fuse together, the fusions of the nucleus of the egg and also the sperm from these 2 organisms. Right? Can you see the difference?
Yeah.
Yeah. All right. So it is very important for you. There are 5 basic steps. Probably they are slightly different, but they go through the similar step. Okay. One is, you can see, is this one is like external fertilizations. The other one is the internal fertilizations. Like mouse, it is internal fertilization, but the sea urchin is the external fertilizations. Anyway, they go through the similar sperm egg recognitions steps. Okay. So meeting between the sperm and also the eggs is not a simple matter. Of course, for example, in the marine organism, they release their gametes into the environment, small as a tide pool and large in the oceans. But moreover, it shared with the other species that may shed their sex cell at the same time. So because for example, in the ocean, there are so many species. There are so many eggs and so many sperm. And how could they recognize each other? Yeah? Have you ever asked this question?
Yes.
Yeah, there are
One.
thousands and thousands and thousands of species of fish in the ocean, right?
Mm.
Yeah, how come they are so specific then?
Mm.
Right? So how can sperm and eggs meet in such a dilute concentration? And how can sperm be prevented from trying to fertilize the egg of other species? Right? So it is the species-specific extractions of the sperm and species-specific sperm activations that will allow them to recognize each other. Right? So the species-specific sperm extraction has been documented in the numerous species, including Nigerians, the mollusks, the echinodons, and also the Eurocodiates. So the sperms are attracted towards the eggs of the species by chemotaxis. Chemotaxis is follow the ingredients of the chemical secreted by the egg. And the mechanism of the chemotaxis is differ among the species, of course. Each of these species, they have different chemotaxis mechanism. And reset, okay, reset is a chemostatic molecule that's isolated from the egg jelly of the sea urchin, which could attract the sperm migrations and is also act as a sperm activating peptide. If you look at this one, yeah, this is A, okay, A is a second photograph was posed showing the sperm swimming in the tight circle before the addition of the reset. You can see can you see the round figures here, the round things here?
Yes.
That is the sperm. This one is not being activated. The reset is not being added to this for example, petri dish, for example. When you add this the reset in this area, so you can see the sperm slowly moving. You can see the white structure, they are slowly become dense here, right?
Yes.
Yes.
Yes, and they become denser at the sea. See here?
Yeah.
And then you can see most of them, they're moving towards this direction. So actually, reset is the molecule that are released by the egg to attract the migrations of the sperm towards the direction of the egg. Yeah, this is one of the sperm chemotaxis mechanism for sea urchin. Boleh faham?
Like a
Boleh.
magnet. The magnet, right? So,
Yeah, it's like a magnet lah. Dia sebenarnya like a inducer. Dia memberikan signal, right? Memberikan satu signal kepada sperm supaya sperm tu boleh migrate ke arah egg itu, telur tu ataupun ovum tu, supaya the sperm boleh migrate kepada egg tu supaya egg tu boleh di-fertilize.
Oh,
okay.
And dan ini adalah species specific. Because other sea urchin punya species pun dia gunakan mekanisme sama, tetapi dia lain. Species yang lain dia tak boleh attract kepada species yang lain. Dia kena attract pada species yang sama. Boleh faham tak?
Boleh.
Okay. So the acrosomal reactions in the sea urchins is that first is the fusion of the acrosomal vehicle with the sperm plasma membrane, which is an exocytosis that result in the release of the content, okay, of the acrosomal vehicle. And the second one is the extension of the acrosomal process, and this is the reactions is initiated by the contact of the sperm with the egg jelly, and this contact causes the exocytosis of the sperm acrosomal vehicle and the release of the proteolytic enzyme that can digest the path through the jelly cord to the egg surface. And we know that the egg jelly is very thick, isn't it?
Yes.
So there will be the acrosomal vehicle. They will release a kinds of chemical, which is a proteolytic enzyme, and this proteolytic enzyme will help to digest the thick layer of the jelly layer, the egg jelly, so that they'll go through the inner part, so that the egg can meet with the sperm. Boleh faham tak?
Boleh.
Okay, contohnya di sini saya show ni sebenarnya. Di sini. Okay, on ini acrosomal reaction kan. So this acrosomal process, it will release kinds of chemical, what we call the proteolytic enzyme, and this proteolytic enzyme will help to digest the jelly layer, so that there will be a binding of the sperm to the ventellin envelope here. And at the end, it will fuse the nucleus. The nucleus will release, so that the nucleus will be released from the sperm and fuse with the nucleus of the egg. Ya, boleh faham tak?
Boleh.
Boleh.
So ini pun acrosomal reactions of the sea urchin. Kita boleh tengok, ya, ini taken by the electron micrograph, right? Dia ni acrosomal enzyme akan di-release, akan dikeluarkan untuk mendigest, untuk mencenarkan the thick jelly layer of the egg. Okay?
Okay.
Right. Ini pun sama. Okay? But okay, satu lagi kita apa yang saya nak attract your attention is that the species specific recognition of the sea urchin. Another molecule is what we call the bindin. Okay, the bindin is the acrosomal proteins that mediating the recognition between the sperm and eggs in the sea urchins. So the biochemical study shows that the bindings is closely related. Sea urchin species are indeed different. For example, we have two difference. Okay, if you look at the S Paperatus and Franciscanus. Okay, kalau kita tengok the bindings particle, ya, untuk spesies yang sama dia akan bind together kan.
Yes.
Tapi kalau kita gunakan yang ini, okay, binding daripada Paperatus, dia tidak akan bind kepada apa tu?
Another species.
Another species, which is a Franciscanus kan.
Yes.
Or vice versa, kalau kita gunakan apa tu binding particle Paperatus untuk bind kepada apa tu Franciscanus ni kepada yang Paperatus, dia pun tidak ada coagulation tidak ada berlaku kan, kerana dia tidak recognize each other.
Yes.
Yes, can you see the difference? Only
Yes.
yeah, only the binding from the what we call the Franciscanus recognize the Franciscanus punya molecule. Right? So ini ada coagulations, ini ada coagulations. But if two different species, there are no coagulation because they are not recognize each other. Dia tidak mengenal. So this is what we call the species specific punya recognitions in the sea urchin. Boleh faham?
Boleh.
Senang saja kan?
Ya.
Okay, apa yang kita nak utarakan di sini ialah kita nak tegaskan bahawa semua processes ini dia adalah species spesifik. Kita harus boleh imagine ya, kerana dalam sungai ataupun tasik ataupun ocean ataupun apa tu laut, especially laut sangat besar. Ya, dan dia terdiri daripada beribu thousands of organisms, and thousands of different species of organisms is in the sea. So how they actually recognize each other, this is very important. Betul tak? Kalau dia tidak ada species specific recognition, akan just imagine what will happen.
The sperm lost their way.
There will be a hybrid, a lot of hybrid. Tidak ada spesifik punya sudah kan? Ya,
Yeah.
ya. And then the egg and the sperm dia tidak kena di mana satu spesies dia. Dia bind sebarang saja. Betul?
Yes.
Yeah.
So kita tengok begitu smart sekali our God by creating all these species specific punya recognitions for the fertilizations to take place and for the continuations of life. Betul lah? Very smart, isn't it?
Yeah.
Yes.
Yes.
Okay, so the gamete binding and recognitions in mammals, kita tengok zonal pellucida in mammals is equivalent to the ventellin envelopes in the invertebrates, and in this matrix is synthesized and secreted by the growing oocyte and play two major roles during fertilization. One, the first one, it binds to the sperm, and second, it initiates the autosomal reactions after the sperm is bound. Okay, bound to the what we call the zonal pellucida. Okay. So another one is the ZP3. ZP3 is the proteins, the sperm binding proteins of the mouse zona pellucida is also a very specific glycoproteins in the mouse zona pellucida to which the sperm binds, and it's also initiate the autosomal reaction after the sperm has been bound to it. Means the sperm has been bound to the egg. Okay, so unlike sea urchins, the autosomal reactions in mammal occur only after the sperm has bound to the zona pellucida, as I mentioned just now at the earlier slide. Okay, and also the acrosomal reaction differences between the sea urchins and also the mouse may due to the thickness of the extracellular envelope surrounding the egg. Ada tak tadi saya ada bagi tahu kamu the thickness is different.
Yes.
Yes, of the extracellular envelope kan. All right. So the fusions of the egg and sperm plasma membrane, this is a very important process because the recognitions of the sperm and also the ventellin envelope and zona pellucida is followed by the lysis of that portions of the envelopes or the zona in the regions of the sperm had by the acrosoma enzyme or the proteolytic enzyme. The lysis is followed by the fusions of the egg's plasma membrane with the plasma membrane of the egg. This is particularly important process, and in sea urchins, all regions of the egg plasma membrane are capable of fusing the sperm. Not applied to every species. Yeah, and the fusogenic protein play a very, very important role in this process, and in sea urchin, the binding play a secondary role while the member which is the fertilins are essential for the sperm membrane egg membrane fusions. So also, although they are play the similar function, but in different animal we term it in different name. Okay, like the binding it is in the sea urchin, but the fertilins is found in the mammal. Okay, although they play the similar function. Yeah. So this is an example of the sea urchin fertilizations. Okay, not much to tell you this. So the preventions of the polypon, you can see that there are so many sperm actually attached to the egg, isn't it?
Yes.
Yes. How? But what are the mechanisms that will assure that there only one sperm can fertilize one ovum or one egg? There must be a mechanism to it, isn't it?
Yes.
Yeah. So as soon as the sperm has entered the egg, it is crucial to assure that only one sperm may fertilize the egg. So there are what we call a scenario which is a monospermy, by which one sperm enter into the eggs, and we could also have the polyspermy, whereby more than one sperm enter the eggs, which lead to the disastrous consequence of the mouse organism if there is no protection. It could lead to the polyspermy. So the species have evolved to prevent the unions of more than two haploid nucleus. Okay, so in sea urchins has two mechanisms to avoid polyspermy, not polyspermy. Poly, yeah, this is why polyspermy. One is what you call the fast reaction. One is called slow reaction. Okay, the fast reaction is an electric change in the egg, and the slow reaction is the exocytosis of the cortical granules. Okay, there are two different reactions to prevent the polyspermy from occurring in the sea urchin's fertilization. So the first block of the polyspermy is by changing the electric potential of the egg plasma membrane. However, this process is transient. Transient, that means sekejap saja, temporary. Okay, so the unfertilized sea urchin egg have a negative charge inside, but the charge become positive upon fertilization. Okay, so the membrane provide a selective barrier between the egg's cytoplasm and the outside environment, and the ionic concentration of the eggs differ greatly from that of its surrounding. So by changing the electric ion charge, so then from negative to positive, which to prevent by a fast reaction, okay, from polyspermy. Okay, how about for the slow block? The egg of the sea urchin develops second mechanism to prevent polyspermy, and this is because the fast block is transient and not efficient or sufficient to prevent the polyspermy. So during the fast block mechanism, polyspermy can still occur because the ventillin envelope is not removed. Okay, but this removal is accomplished by the cortical granule reactions, a slower mechanical block to the polyspermy that becomes active about a minute after the first successful sperm egg attachment. See, how can this occur is that you can see there are so many sperm that are attached to it, isn't it? Correct?
Yes.
Yeah,
Yes.
but slowly after the entry, one sperm successfully enter the egg, then slowly this layer, the sperm which is attached to the egg, will slowly remove. Yeah, at the end in the D here, the fertilization envelope is completely and excess sperm has been successfully removed. Okay, nampak tak ni?
Nampak.
Okay.
Yeah.
So bagaimana dia berlaku ialah kalau kita tengok ya, banyak sperm akan attach kepada the ventillin envelope. Yes, there are thousands of sperm attached to the ventillin envelope. But once sperm successfully enter the eggs and then do the fusions, so you can see these and the ventillin envelope di sini dia ada banyak spike ni kan, dia akan menjadi licin. Ya, dia punya permukaan ni, yang permukaan macam ni dia menyenangkan sperm untuk attach kepada sperm, betul tak? It's easy the sperm to attach to the ventillin layer because there are more surface for the sperm to attach to, isn't it?
Yes.
Yes. So once the cortical granules containing enzyme, which is the orange and also the green, then this is the actin microfilaments. Okay, this one akan menyebabkan dia punya permukaan di sini menjadi licin. One menjadi licin, maka sperm is very difficult to attach to the ventillin layer. And this is how the excess sperm been removed.
Alright.
Alright, boleh faham? So this is a slow block punya reaction. Berbanding dengan yang fast block, di mana daripada negative charge dia menjadi positive charge. So there are two mechanisms, which is a fast block and slow block of the polyspermy. Boleh faham ya?
Boleh.
Boleh.
Okay.
Recording stopped.
So itu saja untuk hari ini. Ada apa-apa soalan? Jelas tak?
Jelas.
Jelas.
Doktor.
Okay.
Jelas, dokter.
Okay. Kalau tak ada apa-apa soalan, then siapa perlukan attendance? Siapa? Aiman,
Ya,
kamu ada? Aiman ada soalan. So Aiman, apa soalan dia? Aiman kata ada soalan kan? You type dekat chat box.
I think he's typing, doctor.
grouping kah? So sebelum tu mari saya biar siapa yang belum scan tu kamu scan dulu. Jadi group 2 siapa belum scan? Group 3 belum ada siapa group 3 yang nak scan?
Ada. Group 3.
Ada group 3.
Okay. Jadi group 3.
Doctor, Aiman sudah type his question in the chat box.
Okay, let me check. Adakah twin are considered polygamy? Hayo, Aiman, you shouldn't. You are not supposed to ask this question. You know why? You sampai sekarang kamu belum tahu tak apa itu twin, bagaimana twin berlaku kah? Apa? Cep. Tak apa, kamu tak faham, biar saya cerita. Di sini ya, sekejap. Okay, Grace, group 2.